All right, so there's one more type of problem that I want to show, and this falls into the category of gene interaction. It's mentioned in chapter 5. They didn't have any good problems in there. So it's this problem here on the course web called the complementation problem. And so I'll just do an example here in chapter 5. So complementation analysis is a genetic technique or a genetic analytic approach that answers one question. So the question is, let's see. The question is, how many genes have been mutated? So in the last example, we saw a situation with some flowers where we had purple or white and 2 genes were involved. And so just to remind you of that particular case, so this is related to that. So if you had, so in this case, we'll say normal is purple, and then you isolate a mutant somehow. In that mutant, you have 2 mutants. The first mutant is white, and the second mutant is white. Now, if you've mutated a gene, let's say, so it says mutant, and so let's say we've mutated this gene, and there's only one gene involved, and we're going to call it W for white. So let's say we've mutated this white gene, and this only works for recessive mutations. So we've isolated a mutant, and we've shown that it's when it's homozygous recessive, it's white. And we've done this twice. So we're going to call this mutant 1, white 1. And we have another one called white 2, which we know is also white. Now maybe there's only one gene for purple. And if we've got 2 different experiments and we've mutated that same gene 2 times. To figure that out, we cross these together. So we'll call this white 1 and white 2. And we cross mutant 1 across to mutant 2. And if we've mutated the same gene, so this guy we know is homozygous for mutation 1. This is homozygous for mutation 2. If these are the same gene, then these 2 things are alleles of each other. And when we do mutant 1 and mutant 2, it's still going to be mutant because there's only one gene involved, and we've mutated both versions of that. And then this is going to be all white. So that makes sense. If we mutate the same gene and we cross it to itself, all we can get out of it is the mutant, right? But what if we mutated a different gene? What if there's 2 genes involved? So let's say we mutate the A gene. So we'll call it A and B now to distinguish between 2 different genes. And that gives us a white phenotype. Let's do a new page for that. So now we've got a white mutant. And we'll just give it an A. And then we've got another white mutant. And we're going to call it little b. And then we cross these guys together. Now, here's where it gets tricky. So if this guy is mutated for gene A, that means it's normal for gene B. So we want to keep that in mind and include that in our cross. If this guy is mutant for gene B, it's going to be normal for gene A. Could be mutated for both, but we did some genetic trickery and we showed that it's only mutated for one gene. So in this case, we cross these together and we'll get a big A and a little a and a big B and a little b. But these guys won't be white anymore. They'll be purple. Because this guy is white because he's mutant for A. This one over here is white because it's mutant for B. but this one's not mutant. It's heterozygous for both. And because those mutations are recessive, normal, so big A gives you purple, big B gives you purple, but you have to have both of those in order to be purple. So you have to have this. So it's one of these situations. And if you get little a, you're white. And you get little b, it's white. And so now if we cross these together, we'd get 9331. And these would be purple. These guys, because they're mutant for A, would be white. These guys, because they're mutant for B, would be white. And these guys, because they're mutant for both, would also be white. And that's because of this story over here. We made another white pigment, and then we required 2 genes in a 2-step process to get from white to purple. And if we're mutant here, oops, that's B. Mutant there, we're blocked and we're white. If we're mutant here, we're blocked and we're white. If we're mutant at both, we're purple or we're white. And the only way to get to purple is if we have both. So what this gives us, the geneticists have used this idea to answer the question. So we got 2 situations here. We get one pattern if there's only one gene involved. We take 2 mutants. So mutant crossed a mutant is still mutant. So that means that's the same gene has been mutated. Here, we've crossed mutant to mutant. So this is a mutant. And this is a mutant. Now we've got purple, which is normal, which is wild type. If we see that pattern, so we can tell this without even doing the 9 to 3 to 3 to 1 thing, just from this part here. So over here, if we cross mutant, cross to mutant, and we get normal, and in this case, purple is normal, that means that the 2 different mutants complement each other. It means there's 2 different genes involved, and those genes complement. So this is what we call complementation. So just to remind, this is what we're talking about, complementation. So complementation means that one thing complements the other, or one thing sort of makes up for the other. So, so the 2 genes, so the 2 mutants complement each other. So the 2 mutants complement each other. And what that means is because there are mutations in different genes. And when you put them together, you make this heterozygote. That's heterozygous for the mutation in each gene. But this mutant, when you made them together, provides a capital B. And this mutant provides a capital A. So by providing a capital B or providing a capital A, you're making up for the mutation in the other one. So this guy on the left, when he provides a B, it makes up for the fact that the other one has a little b. And this guy on the right, when he provides a big A, it makes up for the fact that the other one is contributing a little a. So big A and little, so the big A complements the little a, and the big B complements the little b by making up for it and providing what's missing. So let's look at that one more way. I think that pretty much covers it. So now let's look at how we can use this. So the way this is used in genetic analysis is to do this kind of an experiment, where you take a bunch of mutants and you cross them together. So in this case, rather than purple mutants or white mutants, we have black mutants. And this is in Drosophila. These different mutants lead to black body when homozygous. So these are all homozygous recessive mutations. And then we cross them all to each other. You might see a word for that where we cross them in every kind combination like this. So we cross, what this table shows is we're crossing B1 to B1 here. So that's black mutant 1 cross to black mutant 1. And this minus means when you do that, you're still mutant. Whenever you do that, whenever you get a minus, that means they're in the same gene. And black 1 and black 1 are the same thing, so they have to be in the same gene. But if we look at black 1 and black 2, we get a plus. Plus means wild type, which means normal. So that means that black 1 complements black 2. And they're in 2 different genes. The mutations are in 2 different genes. What does that mean? That means we have some kind of black pigment that gets converted to some other pigment that then gets converted to normal pigment, which is tan in fruit flies. And this might be the black 1 gene and the black 2 gene. And we need at least one normal copy of black 1 and at least one normal copy of black 2. We need both of those to get normal. If we mutate black 1 and we have 2 mutant copies, then it's black. If we mutate black 2 and we have 2 mutant copies of black 2, then it's black. But when they complement, it's because we're making this guy heterozygous for a normal version of its gene, and this one heterozygous for a normal version of its gene. So it would be like this is that if that was the A gene and it would block it there, and this was the B gene and it blocked it there, when we cross them together, we end up with heterozygotes at both genes, and then it goes normal. So this is just another way to write heterozygous. So that's a lot of stuff, but all of that is there to just say that, just remember that if you cross 2 mutants together and they're still mutant, that means you've mutated the same gene. If you cross them both together and they're wild type, that means you've mutated different genes. If they do complement, that means they've been mutated in different genes. So we can use this analysis. So we've got 7 different mutants. We can say, well, how many genes have we mutated? Have we mutated the same gene 7 times? Have we mutated 7 genes and just mutated each one once? Or have we mutated 3 genes and we mutated one of them twice, the other one twice, and the other one once? Something like that. So how do we analyze this? It's pretty easy to analyze, but you just have to remember, you just have to do it right. It's easy to do correctly, but it's also easy to mess it up. So the way you do this is you group the minuses together. That means, because we said if you cross 2 mutants together and you're still mutant, that means they're the same gene. So 1 does not complement 4, and it does not complement 6. So that means 1, 4, and 6 go together. That means they're the same gene. They're different mutations, but they're mutating the same gene. And we call that a complementation group. So we could call this complementation group A. And then 2, nothing should ever complement itself. And then 2 doesn't complement 5. So 2 and 5 go together. And so we could call that complementation group B. 3 does not complement itself, or 7. So 3 and 7 go together. And we got them all covered now. And then the rest of them should be consistent. So 4 doesn't complement 1, doesn't complement itself, doesn't complement 6. So 1, 4, and 6 make sense. 5 and 5, or 5 and 2 and 5 go together. And 6 goes with 1, 4, and 6. So that's just like this. And 3 and 7 go together. So we sort them into complementation groups. That's what we just did here. How many genes have been mutated? 3 different genes. So a complementation group is the same as a gene. So we have 3 complementation groups, or in other words, we have 3 genes that we've mutated. Now the last thing it says here is how many alleles have been generated in each gene. So we've got 3 genes. We call them C, G, A, B, and C. We've got 3 separate mutations in that gene. So that's 3 alleles. Because an allele is just a different mutation of the same gene. And each of these mutations has been generated independently. So we got 3 in that one, 2 in that one, and 2 in that one. And that's how these problems always look. You're given an experiment, you're given what's called a complementation table. And then you look at them and say, well, which ones do not complement? If they do not complement, they go in the same group. If they go in the same group, they're said to be in the same complementation group. That means they're mutations in the same gene. And however many of those you have, that's how many alleles you have in that gene. Sometimes you might see something like this, where let's say we have an 8th one, and it complements everything. So if it complements something, it means it's different than that thing. So let's say number 8 complements everything except itself. So it complements everything, 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 except itself. So that would mean that we have a 4th gene with only 1 allele, because we've only got 1 mutation that is in that gene. It's different than all the others because it complements all the others, but it can't be different than itself, right? Because that's kind of the definition of being yourself, is you got to be yourself. It can't be different. So if there was an 8th one, then we would say there are 4 genes, 3 alleles, 2 alleles, 2 alleles, 1 allele for the last one. All right, so I'm going to leave that. That's called complementation analysis. The whole point of it is to see if you've mutated the same gene more than once, or if you've mutated a bunch of genes that all give you the same phenotype. So once again, you start with the same phenotype. Start with a bunch of mutants that give you the same phenotype, same mutant phenotype. So in this case, black body. And is each of these mutants, are they in the same or different genes? And if they do not complement, this is where it gets kind of a little bit like reverse thinking. If they do not complement, so if you get a minus, then they're in the same gene. And then different mutations in the same gene are called alleles. All right, so that's complementation analysis.